All right, guys, welcome back to part two. Apparently, my technology is all just taking a dump.
We need an IT guy. So yeah, if you want to work for Bios Life Media, you're an IT dude. Please assist us.
Yeah,
we don't know shit about tech.
You can really help me out because I am like computer illiterate and my phone's falling apart. My computer doesn't want to
Laptop
record
blew up
properly.
and all that kind of stuff.
Oh, jeez. Hopefully, the IT guy tomorrow can help me.
So as Dave was talking about before,
So
our
instead of
tech
just
took a shit.
focal adhesion kinase just being in the focal adhesions of cells where the cell connects to the extracellular matrix or the outside environment of the cell, or the basal membrane with collagen and fibronectin and all those fun things, vimentin. It can also translocate to the nucleus and act as a transcription factor, which is something that Lee is actually much more.
It modulates other transcription factors. Yeah.
It can act in part with it. It can actually phosphorylate June, phos-June, I know that in particular. It can actually phosphorylate GATA and cause its down regulation in terms of degradation.
Right.
I know that was in the VCAM pathway. And
I kind of
it can actually
localize.
has a little something to do with regulating cytokines and basically immunomodulatory function. So
Oh yeah, there was an interesting thing about that. Pretty much when your FAC isn't working correctly, your tumor cell will use that to increase certain signaling that causes T regulatory cells to localize around the tumor and produce a suppressive immune environment, which, for all intents and purposes, helps tumor cells not die.
And grow and proliferate and then metastasize. And it actually causes the CD8 cytotoxic T cells to become overworked and basically not function anymore. And that's actually something that I might be looking at as my
it's a lot of
thesis project.
It's a cool immunomodulation technique. I mean, not technique, but it's the technique that the cells are using essentially.
But
it's definitely Treg mediated, but it starts, ultimately the pathway starts with focal adhesion kinase activity.
Yeah, and I like focal adhesion kinase. It's a nice little protein.
It's the baby right there, man.
It's my dude. It's my dude. All right. So I don't know. I think that's enough of my little spiel. I know you got to get through yours, and we're already at 50-some minutes. So,
well, let's talk about this stuff real quick. So pretty much the stuff that I study is in the big picture, right? All cell types, all cells have DNA.
Do they?
What? Yeah.
They sure?
They do.
I never heard of that.
I'm
pretty sure.
Google it, dude. All cells have DNA, and pretty much what separates your ass from your elbow isn't the location.
Literally.
What literally separates your asshole from your elbow is the type of genetic information being assessed at that moment.
Or the old signals and all the other old.
Yeah, from a developmental context. But I mean, even with that said, if I wanted to reprogram your asshole cells and try to turn them into an elbow,
Good luck.
It's possible. We do it to skin cells all the time. We turn skin cells into neurons all the time.
Pseudoneurons. Come on.
It's a proof of concept. It's a proof of concept type of thing. If you modulate the genetic information that's being assessed, you can modulate what the cell is. Of course.
There's always caveats to that.
Oh yeah, because it's not perfect, because we can't turn off everything.
Exactly.
We do a good job of turning stuff on. We don't do such a great job of turning stuff off.
It's kind of like knowing, Donald Trump has that urine video from Russia, and then being told to just forget about it. I don't think it's going to happen.
I don't know if that's an applicable reference, but
I need to plug that one in there.
Yeah. So back to understanding transcription factors and regulatory sequences. Transcription factors are proteins that bind to DNA, and when they bind to DNA, they modulate the activity associated with genes. They can either enhance the expression, or produce, or tell the gene to be turned on, or they can repress the gene, telling that gene not to be turned on. Right? And how these transcription factors are mediated is through the actual DNA sequences that they bind to. There's also protein interactions that I'll get into later. So these are all of the ways in which you can modulate the production of output, essentially, right? If a protein is going to be produced at the end product, how can you find ways to control that? I principally focused on that area over there called transcriptional control. That's my baby. So when you go from the next slide, right, we go from transcription to control. This is a rudimentary diagram associated with what transcription looks like. We like to call this like the pre-initiation complex, but essentially every gene has a promoter. The promoter is where the proteins bind that tell genes to be turned on.
Mm-hmm.
A bunch of proteins bind to that. DNA loops upon itself, pretty much stabilizing this.
Through mediator?
Through the mediator. Actually, the mediator is a giant protein complex. Yeah, that has like 40-some proteins in it. But press get some ooh, look at this. We get some animations here real quick. So these green sequences associated with the DNA looping are called enhancers.
Mm-hmm.
And enhancers are regulatory elements that bind.
Cis regulatory.
They're cis regulatory elements, yes.
Which means something that works on itself. So
Yeah.
it's one chromosome working on its own self.
Yeah, well, essentially, if you had a protein that was produced in a different chromosome that acted on another chromosome, we call it a trans.
Yeah, that's what
Right.
kind of what I was.
Right, it's games you're alluding to. Yeah, sorry. I'm hyped up for my talk now.
I know, hey man, I like facts, so.
Yeah. So you have regulatory sequences. Transcription factors bind to these regulatory sequences as a whole.
Keep an eye on that.
And when this binding occurs, you get plus or minus transcriptional activity. Let me go to the next slide there.
Boop, boop.
Boop, boop. All right, so this diagram is probably not that visible where you guys are looking from, but when we post it, the explanation will make more sense. So if you have a big long piece of DNA and you have a promoter, you will have different enhancers kind of sprinkled around the genome.
Mm-hmm.
And depending on what proteins, what transcription factors are available at that moment, they will bind these enhancers, and you will get this kind of folding and recapitulation of the DNA molecule that will express.
Certain repressors block certain enhancers.
Right.
And like if you have 1 through 5, it'll make one set of cells here do one thing, and then the area 2 will block 1, and 3 through 5, and then you'll have 2, and then
So you gotta remember, there's definitely this symphony associated with regulatory sequences and the proteins, because every cell has the same genetic information, right? So if you have to make cells different, you need to be able to turn or activate certain areas of the genome on at different points in time based on the proteins.
Mm-hmm.
And so one regulatory sequence, one enhancer, can actually be present in 2 different cell types, but the protein that's interacting with that enhancer may be different. And because that protein is different, what that protein modulates or affects is different within that cell. And the reason why I study this in the context that I am now is because transcription factors are commonly mutated or overexpressed in tumor cells and cancer cells. And when
particularly, I think you're going to talk about one?
I'm going to talk about Burkitt's lymphoma.
Nice. And what would be that protein of interest there?
My boy Mick, my boy C-Mick. Actually, there's a bunch of Mick family members, L-Mick, N-Mick, but I like C-Mick
I actually have to
specifically.
give a journal club article on N-Mick.
Yeah, N-Mick, which is called neural Mick. It's principally expressed in developmental neurons.
I'm actually going to be doing it in the context of neuroendocrine tumors, so that actually is pretty.
It's also, yeah, I mean, in a normal case, N-Mick it's very lowly expressed or not expressed at all in most cell types, but of course, in the context of cancer, things get expressed when they're not supposed to, and shitstorms.
I like how MYC can just be overexpressed and not mutated in
Well,
terms of like cancer
No,
progression.
no, there are MYC mutants.
No,
Yeah.
I get that, but there's a subset that you don't actually have to have a mutation in there.
No, yeah, so
you just
for
have
example,
an overexpression.
yeah.
For whatever reason, it actually drives cancer.
So in Burkitt's lymphoma, for example, you get a translocation of c-MYC into an area of the genome that gets largely expressed, right? So the promoter that is associated with making antibodies, which antibodies are highly expressed because you have to make a lot of them, you get this translocation of c-MYC onto that promoter. And when that happens, MYC gets turned on at a level that is never before seen in any cell type. And because MYC, the concentration of MYC is so high, the cell becomes cancerous. And that alone is enough to turn cells into tumor cells. Aside from its ability to bind to DNA, it also pretty much turns on genes that regulate stability, which is pretty much how stable the genetic information is from a actual physical point of view. If it's less stable, then there can be more breaks. More breaks means more damage. More damage means more mutation. You also are turning on like biosynthetic pathways that allow the cell to make more stuff. Of course, cancer cells need to make more stuff and need to be more energy confident so that they can divide. So yeah, make super important, all this stuff. But to get it like real short with it, my last slide.
Yeah, real short.
Real
with it.
short.
I love it. It's quiet the entire episode. All of a sudden, he just lights up like a Christmas tree when it comes to Mick. Oh,
I
hate Mick.
This last slide is
like
an experimental.
Guy. That's Mick.
Yeah, you're right. Yeah, this last slide is an experimental technique slide. There's 3 things on here: ChIP-seq, in-chip, and MS, which doesn't stand for multiple sclerosis, stands for mass spectroscopy.
Really?
Yeah.
Yeah.
And 3C, which stands for chromatin conformation capture.
So what does ChIP actually mean?
ChIP stands for chromatin immunoprecipitation. Pretty much.
Just give it a real quick overview of what that is.
When proteins bind to DNA, you can use formaldehyde to cross-link them, and then you can use an antibody to extract that protein as it's bound to DNA after sonication. And
sonication?
Yeah, using sound. Yeah, using sound to break DNA. Pretty much. And so because you can use an antibody, a selective antibody, to select for a specific protein, you can then sequence the DNA that comes down with that specific protein, right? And that's ChIP-seq. In-chip MS is actually the utilization of a protein endonuclease called CRISPR, or Cas9 rather. We like CRISPR.
We talked about CRISPR 2 weeks or 3 weeks ago.
Yeah, it's a genome editing technique, but what happens if you create a mutation in Cas9, you can deactivate its endonuclease activity. Mm-hmm. So pretty much if I wanted to know at some location, let's say the focal adhesion kinase promoter, if I wanted to know what molecules are associated with turning FAC on,
turning FAC on.
Yeah, if we wanted to know what that was, I would create.
Phrasing.
Yeah, I would develop an experiment utilizing this in-chip MS technology to pretty much take that entire region of the genomic information as well as the proteins, use something called mass spectroscopy, which tells me what proteins are there based on their size or their amino acid composition.
Yep.
It's a way of looking at areas of the genome where we actually don't know what's going on at a protein level.
Nice.
3C is super duper complicated. If I can pull this off, I'll probably get a Nobel.
God bless you, man.
Yeah, people do it all the time. It's super hard to do, but pretty much you can use it to look at what enhancers, those regulatory sequences I mentioned earlier, are associated with a specific location in the genome at a given time.
Nice. Sounds like a lot of work.
Sounds like a lot of work, yeah.
Sounds like a good thesis project, though.
Yeah, I like it.
Nice. All right, guys. I know we're well over time. We're for like an hour right now. Thanks for sticking with us. Thanks for sticking with us for the second video. We'll have to throw up both of them on the website.
Yeah.
Next week, we'll be talking about what is cancer.
That's my excitement.
Trust me, we'll be a little bit more prepared. We'll have less technical issues. We'll
we'll try to like work those out like today.
Yeah, tomorrow, basically. I have to go to the IT guy and hopefully, if anybody knows, just let me know. I'll listen to your advice on how to fix my microphone.
Yeah. Outside of that, I also wanted to mention that we're doing some grad student interviews. We just published one today. Shout out to Martina at the University of Basel in Switzerland. Or
yes.
Yeah, pretty much. Yeah, she's a fourth year PhD student in chemistry.
B-A-S-I-L-E?
What? Basel? Basel? B-A-S-I-L-E? Yeah. Basile. Yeah, excuse my, I'm not cultured at all. I'm from Brooklyn. I don't even. But yeah, we're interviewing grad students.
I'm cultured? Are you kidding me?
There's a smart cookie over here. We're interviewing grad students in science to pretty much highlight you guys and talk about your research. So if you're interested in doing an interview with us, please reach out to us. We'll be happy to interview you and put your research out there.
Yeah, please do. Any type of STEM field. I know that's what
I mean, engineering. I mean, you name it, we got it.
But even like if you do some anthropology, I'm into anthropology.
Yeah, I mean, this is science.
Yeah, it's humanities, but whatever. So next week we're gonna do what is cancer. Tell you why there is no cure for cancer. And really give you an overall point of view. Basically, maybe even talk a little bit more about our research and the respective cancers that we're going to be looking at.
Unless you have Philadelphia chromosome positive leukemia, then we kind of had a cure for that.
Yeah, well, there's a lot of some lymphomas do have some cures. But yeah, other than that, guys, thanks for tuning in, and we'll see you next week. All right, bye bye.